Now we're going to talk about the Crossbridge Cycle. And instead of me reading through this slide, I think it's easier to actually play the video, the animation, so that you can see the Crossbridge Cycle happening. And I'll pause the video and elaborate on some points. But reading through the steps without visualizing it, I think visualizing it first really helps. But there is a slide with it written out. There's this diagram from your book that shows you the Crossbridge Cycle so that if you want to actually see kind of step by step. And then we have these wonderful animations which I will now play, and we'll discuss what the Crossbridge Cycle is using this animation. And the Crossbridge Cycle will begin once calcium is bound to troponin. Troponin has changed its shape, shifted tropomyosin out of the way, and when tropomyosin gets shifted out of the way, remember now these actin active sites are exposed, and that is where these myosin heads, remember these red filaments with the globular heads, that is myosin, or the thick filaments. And these globular heads will bind to those actin active sites. So let's visualize that.
Once the active sites on actin have been exposed, the next important step of muscle contraction is the Crossbridge cycle. During each Crossbridge cycle, the myosin heads bind to actin filaments, pull on them, and then detach. Now let's examine the individual steps of this cycle. First, the myosin head splits a molecule of ATP that has previously bound to it into ADP and a phosphate.
So ATP, which is going to be supplied by all of those mitochondria that are nearby. Remember, there are lots of mitochondria that surround these myofibrils. And so ATP is supplied by the mitochondria, which the mitochondria are constantly undergoing cellular respiration, oxidative cellular respiration, and supplying ATP. ATP binds, and remember ATP is like the primary energy molecule of all of the cells. ATP binds to the globular head, and when it gets hydrolyzed meaning split, cleaved into, so ATP is adenosine triphosphate, 3 phosphates attached. When it gets hydrolyzed, it's hydrolyzed into ADP, which is adenosine diphosphate, 2, and a phosphate. That cleavage, that hydrolysis releases energy, and that energy is what's going to fuel this cross-bridge formation, okay? So that energy that gets released when hydrolysis or the splitting of that bond happens, that's the energy that drives this process.
It uses the energy released to assume a cocked position, much like a loaded spring. All of this actually happens before the active sites on actin are exposed, so once they are available, the now cocked myosin heads are ready to bind to them. Next, a cocked myosin head binds to an active site on actin, forming a structure known as a crossbridge. Note that ADP and phosphate are still attached to the myosin head at this point. As the ADP...
So that video just stated is that the myosin heads are already activated and they're waiting until but they can't really do anything until these actin active sites are exposed. So before contraction actually happens, a lot of these myosin heads will already be quote unquote in the cocked position, cocked and ready to go, okay? So they have already hydrolyzed ATP into ADP and phosphate. They're in the cocked position, and then as soon as these actin active sites are exposed, because calcium binds to troponin, then myosin will bind to actin.
Attached to the myosin head at this point. As the ADP and phosphate detach from the myosin head, the energy stored in the cocked head is released. As it relaxes, the myosin head pivots and pulls on actin. This movement is known as the power stroke. When
ADP and phosphate leave, now that energy stored here also gets released, and the head goes into what's called the power stroke. And that power stroke slides the actin filament in towards that midline, that M line.
After the power stroke is complete, a new ATP molecule binds to the myosin head and causes it to let go of actin. Note that as the myosin heads detach, the actin does not slide back. This is because other myosin heads are bound to actin and are undergoing power strokes.
When ATP binds to the myosin head, then it detaches. So questions I like to ask is, what would happen, or what happens when ATP binds to the myosin heads? You would detaches from actin. What happens when ATP gets hydrolyzed into ADP and phosphate on the myosin head? You would say that it goes into the cocked position, something like that, okay? And I actually think the next video is a lot better, so we'll watch another video.
Not slide back. This is because other myosin heads are bound to actin and are undergoing power strokes.
So here's our sarcomere from Z line to Z line. Remember, we talked about the anatomy of the sarcomere. Here are our actin filaments. Here are the thick myosin filaments. Here's our M line. This pink zone would be the H zone, where you only see thick. From here to here would be our A band, where we have overlapping thick and thin. From here to here would be our I band, where we only have thin. We already went over this in the previous videos, okay? So if you look, these myosin heads that go branch above and below, the way that they're positioned, their power strokes, so on this side, the power stroke would be in towards the M line this way. Over here, these myosin heads, their power stroke would be in towards the M line this way. So these actin filaments are being pulled, and they slide across the myosin filaments in towards, so the myosin filaments stay stable. They stay where they are. They're stationary. But the actin filaments get slid in towards the M line from both directions, and that's how the sarcomere shorten. Therefore, that's how all of the myofibrils shorten, because sarcomeres are just repeated down the length of the entire myofibril. And that's how the skeletal muscle cell shortens during contraction, okay? But if you look, the myosin filaments, they don't change length, and they are stationary. The actin filaments, they also don't change length. They stay the same length the entire time, but they're being slid in towards the M line, so the overall length of the sarcomere shortens, but the filaments do not. The myosin nor the actin get shorter. Just the sarcomere gets shorter because the actin slides in towards the M line. How does it slide in? Because those myosin heads are undergoing this crossbridge cycle several times during contraction.
To summarize, a muscle contraction is simply a succession of crossbridge cycles. For an average muscle contraction, the cycle will repeat about 20 to 40 times in each sarcomere of the myofibril. At any given time during a contraction, some of the myosin heads are pulling on actin while others are detaching and being reactivated by ATP.
Same length, just the actin filaments
myosin heads
are
work
being
much
slid
like
in.
a team pulling on a rope. As the actin filaments slide toward the M line and the sarcomere shortens, the entire muscle shortens and generates tension.
All right, so the contraction cycle may repeat as long as the stimulus to contract continues. So as long as there's a stimulus, contraction will continue. As long as calcium is bound to troponin and there's ATP available, that crossbridge cycle, that contraction cycle will continue. All right? And you can, the average contraction, that crossbridge cycle, like the video stated, 20 to 40 times for each myosin head in each sarcomere. And there are lots of myosin heads in one sarcomere, then there are lots of sarcomeres, and there are lots of myofibrils. So there are lots and lots and lots of these cross-bridge cycles happening during contraction. This is an image. It's hard to see on here because it's so small, but it is an image on your book, and it helps kind of break it down so you can see it has the actin numbered so that you can see when the myosin head binds and undergoes power stroke and then detaches and then goes back into the cocked position and binds and then power stroke. It just gives you a numbered actin subunit so that you can visualize that, when it attaches to 2 and slides it in this way, when it goes back to the cocked position, it will then attach to 3 the next time, and so on and so forth. If you want to look at this in your text, this diagram may help you visualize it as well. So there are lots of good images in your book, but I like the videos. So let's go through the Crossbridge cycle again, and I like this video a lot.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the Crossbridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge.
So the activated myosin head. So what's already happened here is ATP has bound to the myosin head. ATP gets hydrolyzed into ADP and P. That hydrolysis supplies the energy to put the myosin head into the cocked position, and as long as the actin active sites are exposed, that activated cocked myosin head can bind, forming a cross bridge.
Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots.
So what causes the power stroke? The pivoting. So that's just the myosin head pivots. That pivoting is called the power stroke, and it is how the actin filament gets slid in towards the M line. So if we have no power stroke, then we have no shortening of the sarcomere, okay? So how does that power stroke happen? It happens once ADP and P are released. So once ADP and phosphate are released, that energy also gets released, and we have the pivoting or the power stroke of the myosin head. That is when that power stroke causes the actin filament to be slid in towards the M line,
sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches.
All right, so the one power stroke from one myosin head is not sufficient to have full contraction. So each myosin head has to detach, go back into the cocked position, reattach, power stroke. It slides it in a little bit further. Then it detaches, and it will go into the cocked position, reattach, power stroke. Detach, cocked, reattach, power stroke. Over and over again, 20 to 40 times, all while sliding the actin filament in this direction, in towards the M line of the sarcomere. Okay? So in order for that crossbridge cycle to happen 20 or 40 times for each myosin head, it has to be able to detach. It detaches when a new ATP binds. So when a new ATP binds, it detaches. That ATP will then be hydrolyzed, releasing energy into the myosin head, and then it goes into the cocked position. So let me rewind a little bit.
Of the sarcomere. Step 3,
Okay.
cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4, reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position.
All right, so how does the myosin head return to the cocked position? Hydrolysis of ATP. When ATP gets hydrolyzed into ADP and phosphate, that energy is going to cause that energy gets released from the bond into the myosin head, and it goes into the cocked position.
As long as the binding sites on actin remain exposed, the crossbridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract.
So I like this because it's showing you all these little myosin heads undergoing the crossbridge cycle over and over and over again, pulling the actin filaments over here in towards the M line in this direction, actin filaments over here in towards the M line in this direction. The myosin filaments aren't getting shorter, the actin filaments aren't getting shorter, they're just sliding past one another, making that H zone smaller, making the I zone smaller. And then, so I hope that makes sense, linking the anatomy of the sarcomere, which we learned in previous videos, to now actual contraction, structure-function relationships, okay?
Sarcomere shortens. This shortening causes the whole muscle to contract. Crossbridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum.
So in order for contraction to stop, the motor neuron stops sending information for contraction. So the motor neuron stops sending signals. So that means we no longer have an action potential along the length of the sarcolemma. And when that happens, calcium will start to be actively transported back into the sarcoplasmic reticulum. And if calcium goes away, well then troponin will change back to its original shape, and tropomyosin slides back over the actin active sites. And myosin, even if ATP is present, myosin can no longer bind if tropomyosin is in the way. Right? So once calcium goes away, this crossbridge cycle ceases to happen.
Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Okay, so let's watch that again.
Transported back into the sarcoplasmic reticulum.
So calcium is being actively transported back to the sarcoplasmic reticulum. No more calcium is present.
Troponin returns to its original shape.
So those little pink molecules that were floating away, those were calcium. Troponin goes back to its original shape,
allowing tropomyosin to glide over and cover the myosin binding site on actin.
When troponin goes back to its original shape, tropomyosin then slides back over those actin active sites, and the myosin heads can no longer bind. This is one of those supplemental videos that I posted earlier on in the beginning of last week, so you can rewatch this as many times as you need to understand the Crossbridge cycle. How I like to ask the Crossbridge cycle is I will say, what happens when ATP binds? And you need to know that when ATP binds to the myosin head, it detaches. What happens when ATP is hydrolyzed to ADP and phosphate? Well, energy is released and it makes the myosin head go into the cocked position, right? What happens when it's in the cocked position? When can the myosin head attach to actin? When calcium is present and tropomyosin exposes the actin active sites. What happens when ADP and P leave or released from the myosin head? That's when you get the power stroke. Something along those lines, okay? Very rarely do I ask you to, just a short answer question, list all of the steps. I'll usually give you something and you tell me what happened. I'll give you one step, you tell me what happens next. So hydrolysis of ATP is responsible for, hydrolysis of ATP, that is when ATP is hydrolyzed into ATP is hydrolyzed into ADP and P, okay? It's hydrolysis of ATP is responsible for release of the myosin head from the actin active sites. That's incorrect. How does the myosin head get detached? It's when ATP is bound, not hydrolysis of ATP, but when a full ATP is bound. Recocking of the myosin heads. Okay, so maybe. The power stroke, well, that's incorrect because the power stroke happens when ADP and P are released. The movement of tropomyosin exposing the actin active sites, well, that's incorrect because the movement of tropomyosin to expose the actin active sites, that is a result of calcium binding to troponin. Ooh. So the correct answer here is the recocking of the myosin heads. This is something similar to how I might ask it on the test. The binding of ATP to myosin is responsible for, so this is like very similar to the types of questions I ask on my exam. The binding of ATP to myosin is responsible for, so when ATP binds to the myosin head, remember it detaches. So it's best if you can know this stuff so well to where you don't even, the answer, you just have to look through and find it in multiple choice. So release of the myosin heads from the actin active sites, okay. Recocking of the myosin heads, no, we just had a question like that. And what's responsible for the recocking of the myosin head? It's the hydrolysis of ATP that provides the energy to cock the myosin head. The power stroke, well, we know that happens, the pivoting of the myosin head happens when ADP and P are released. The movement of tropomyosin exposing the actin active sites, no, that's a result of calcium binding troponin. So the answer here is when ATP is bound to myosin, it detaches the myosin head from the actin active sites. The release of ADP and P from the myosin occurs during the power stroke. All right, so I just went over these types of multiple choice are very similar to how I might ask it, or I may ask it in short answer form where I give you this and I want you to tell me what happens immediately after, like what happens next. But it wouldn't be like list all of the events of the Crossbridge cycle. The myosin heads return to their low energy, relaxed state during, so the myosin heads return to their low energy, relaxed state during the power stroke. This, I probably wouldn't ask something like this, but the myosin head returns to their low energy, relaxed state during the power stroke. Think about it this way. When the power stroke happens, ADP and P are released, and that energy is also released. I said that multiple times, right? When ADP and P are released, the energy is also released, and that causes the power stroke. Well, if energy is released, that means it would be in a low energy, relaxed state. Then another ATP binds, it detaches, that ATP gets hydrolyzed, it becomes charged, and in the cocked position. So the cocked position of the myosin head is its high energy form, okay? So that's how you could have thought about this logically through what you do know. The power stroke, all right, so pulls the thick filaments towards the Z lines. Oh, wait, are thick filaments being pulled during contraction? No, it's the thin filaments. And then towards the Z lines, we know that the actin filaments are slid in towards the M line. The Z lines are the delineating from sarcomere to, it delineates what a sarcomere is, right? A sarcomere is from Z line to Z line. Okay, so positions the myosin heads in their high energy position, shortens the length of the thin filaments, pulls the thin filaments towards the M lines. Ah, there we go. Shortens the length of the thin filaments. I said this multiple times. Do we actually shorten myosin or do we shorten actin? No, those filaments stay the same length, but the sarcomere shortens because the thin filaments, the actin filaments, are pulled in towards the M line, but they're the same length. So rewatch those videos if you need to see that visually. But the actual length of these filaments do not change. Only the length of the sarcomere changes. And actin is always pulled in towards the M line, which M line, the middle line, right down the middle of the sarcomere. This is just a little, if you want to read through this PowerPoint slide, it just gives you kind of an analogy of the sliding filament mechanism, and it uses sailors pulling on an anchored rope to help you understand it. So if you want to read through this, if you're having trouble understanding or visualizing the sliding filament mechanism of actin being slid over the myosin in towards the M line. And in the next video, I'll talk about skeletal muscle relaxation.